Now, let's shine the spotlight on actin filaments, or what we call microfilaments. They're not just ordinary filaments. They have a flair for polarity, just like microtubules. This means that their polymerization or assembly and depolymerization or disassembly occur more rapidly, and preferentially at one end compared to the other. The end where polymerization occurs faster is called the plus end or barbed end, while the end where depolymerization occurs slower is known as the minus end or pointed end. So, it's like the plus end is the VIP section of the actin filament party. But, can you guess? What gives actin filaments their polarized look? It's all thanks to the asymmetrical subunits aligning themselves along the filament. They're like synchronized dancers, all pointing in the same direction, forming a captivating double helix. The subunits in the two strands stagger themselves, creating a mesmerizing pattern of asymmetrical arrowhead seen when myosin heads bind along the length of actin filaments. It's an artistic masterpiece. Here's where the plot thickens. When free G-actin subunits, the solo performers, come into play, they bring their partner, ATP, with them. And guess what? When they join the growing actin filament, they become enzymatic magicians, hydrolyzing ADP and releasing a burst of energy. The resulting ADP finds its place deep in the groove of the filament. And like a magic trick, the filament lengthens and elongates. And here's the twist. As the actin filament elongates, the tempo of ADP dephosphorylation quickens, adding a touch of excitement to the performance. But what happens when the G-actin monomers exit the stage from the minus end? They've got a surprise for you. When G-actin monomers are removed from the minus end of the filament, they carry ADP molecules attached to them. And once they step into the cytosol, ADP molecules gets phosphorylated back to ADP. It's like a molecular rejuvenation, preparing the actin monomers for another round of polymerization. But wait, there's more. It's not just about ADP and ADP, but also about the intricate dance between actin and various actin-binding proteins that interact with both G-actin and F-actin. These actin-binding proteins have the ability to control the lengthening, shortening, and even branching of actin filaments. They can accelerate or decelerate the filament's growth and control its structural changes. During the formation of actin filaments, the action is happening at both ends. Actin monomers eagerly join the plus end, fueling the growth of the filament, while, simultaneously, some monomers bid their farewell from the minus end. This dynamic interplay, known as treadmilling, creates a harmonious equilibrium, where the number of actin monomers added to the plus end matches those being removed from the minus end. So, there you have it. A captivating tale of actin, the protein superstar. From its abundant presence in cells, to its intricate structure, and fascinating interactions with nucleotides, actin knows how to steal the show. Whether it's in muscle cells, showcasing its contractile abilities, or in non-muscle cells, maintaining structures, and orchestrating movement, actin knows how to perform. So, next time you witness a mesmerizing cellular performance, remember that actin is the star of the show, bringing elegance, dynamics, and versatility to the grand stage of life.
From medical students to seasoned professionals, sqadia.com offers engaging and dynamic content for your educational journey.